Thanks. So yeah, hi everyone, I'm Laura, I'm a PhD student, and yeah, as mentioned, my research is generally looking at the dietary responses of European deer in relation to climate change in the Pleistocene. And today I'm going to speak about the paleoecology of late Pleistocene European deer using the method of dental wear analysis. So when I tell people that I'm studying the diet of deer in the Pleistocene, people do usually ask, why deer? Well, although people think that deer are very common today, they are actually a taxa under some stress. 66% of them have a declining population trend. In the Pleistocene fossil record, however, they are relatively numerous compared to other species, which makes them a great study taxa. Also, different deer species live sympatrically at the same sites, which we'll see examples of shortly. And this makes it easier to study interactions between the species. They also have fairly wide habitat preferences, from reindeer out in colder habitats to roe deer needing warmer forested habitats. This means they're present in many different periods and environments of the Pleistocene. And then lastly, most species I'm studying today are still alive on Earth today, making any results I get more applicable to the species today. So here are some of the species that I'm studying for my PhD, but today we're just going to focus on these three. So it's reindeer, red deer, and giant deer, which disappeared from northwestern Europe about 13,000 years ago and finally became extinct around 7,500 years ago. So the Pleistocene is a very useful study period because it has well-defined warm interglacial periods and cool glacial periods, which allows us to study species responses to different climates. In Britain, since marine isotope stage 12, the melting of the Anglian glacial caused the previously permanent land bridge connecting Britain to mainland Europe to be cut off by a water channel. Then with each glacial period, the increase in ice levels meant that seawater levels were reduced and the land bridge reappeared. And then with the following warm period, the melting of the ice increased sea levels again, meaning that the land bridge was submerged again. This means that species could only migrate in and out of Britain during certain climates, which today produces well-established biostratigraphically indicative assemblages of mammals for each interglacial after stage 12. So the two techniques which make up the method of dental wear analysis are mesowear and microwear. They each give an indication of an individual animal's life on different timescales. So mesowear is a longer term signal of diet, capturing the diet over sort of months to years of the animal's life. It uses the sharpness of the tooth cusp to determine what the animal was eating. So vegetation such as grasses have a more abrasive component in them and wear the tooth down during chewing food, producing a blunter cusp, as you can see on the right. However, softer foods like leaves maintain a sharper tooth cusp, such as this one on the left. With microwear, we can see diet on a much shorter time scale of the individual's life, so it's sort of down to the last few meals before it died. And then looking at casts made of individual teeth under the microscope, the chewing surface has different micro scars on it in the form of scratches on the left, which are indicative of more abrasive foods cutting across the tooth surface, and pits on the right, which indicate softer foods. And then it's the relative quantities of these scars that indicate a certain diet. And these methods are really useful to combine as it can help understand the flexibility of an individual's diet over the course of its life. But today we're just going to focus on microwear, so it's the short-term indications of diet. So for this study, I focus on these three species, red deer, reindeer, and giant deer, and counted the number of scratches and the number of pits in two small areas of their teeth and took an average for each individual. I then worked out a scratch to pit ratio for each individual in order to easily compare results. And then the higher the number of this ratio, the more of an abrasive diet the individual had, which can be related to higher consumptions of grass. I looked at diet over these 7 sites, most in England and one in southern France, and they're all in marine isotope stage 3, so between 60 and 30,000 years ago. So these are the results of the study. Each site is along the x-axis, and up the y-axis is the scratch pit ratio. As previously mentioned, the higher the ratio, the more abrasive was the diet. You can see that reindeer, the blue dot, had quite a variable diet, but potentially increasing the abrasive components such as grass when sharing a site with a higher number of other cervid species towards the right of the graph. At the French site of Gargas, giant deer have a strong browsing signal. While reindeer also have a browsing signal, it seems they've taken more abrasive food items than giant deer. The position of Gargas Cave in the Pyrenees is in the valley of the Nest River and its confluence with the Gargonne. We think that diverse ecological niches surrounded the area, including both mountainous and plains ecosystems. So it might be that reindeer incorporated more abrasive foods, such as the grass on these plains, in order to niche partition from the browsing deer. However, perhaps the most interesting result here is the stronger abrasive signal at Kent's Cavern for red deer and giant deer. At this site, their body size is noted as much larger than average, especially for red deer, which increased by 37% compared to the Pleistocene average. Giant deer body size increased by just under 100 kilograms, which is about a 17% increase. However, at two very geographically close sites to Kent's Cavern, which are Gully Cave and Wicky Hole, the red deer diet shows a much less abrasive signal. So it seems that something about Kent's Cavern specifically was causing them to supplement their diet with abrasive foods such as grasses, which are seen as poorer quality food compared to browse. Another thing to note is that when we look at the same results but add in all of the individual points, so this is the scratch pit ratio for each individual deer I studied, in addition to the average for each species at each site. We can see that in general, but especially at Gargas and Pinhole, reindeer and giant deer have very good separation between the abrasivity of foods they consume. As you can see, they don't really have any overlap in their individual points of scratch pit ratio, which could translate into dietary niche differentiation. But as you can see at Kent's Cavern, all of the deer species, but especially red deer, have a very wide spread of scratch pit ratios in the individuals I counted, indicating very wide dietary niches for each species. So it could be that this increase in grazing signal at Kent's Cavern for red deer and giant deer is due to the increase in body size, and as larger body sizes produce higher metabolic demands, larger quantities of food are needed to consume enough energy. Eating more grass, which is seen as less nutritious but more abundant than browse, will help fulfill this energetic demand. It could also potentially be for niche differentiation between large deer. For example, in giant deer at the same site as red deer, they seem to incorporate more grass to widen their dietary niche to lessen the overlap with red deer. And as you can see at Pinhole, red deer are not present. Giant deer have more of a browsing signal at Pinhole when red deer are not present. During stage 3, red deer have a northern limit to their range in the UK, and Pinhole is in northern England. So we know that red deer were not present at this site. Therefore, it could be that giant deer do not need to take in more grass in order to niche partition with red deer, allowing them to maintain more of a nutritious browsing diet. Lastly, it could be a seasonality signal, with deer dying all year round at Kent's Cavern and showing a wider range of food abrasivity from different seasons, but perhaps only being taken seasonally at sites such as Gargas and Pinhole. So while there is still much future work to do to untangle these different signals and add more data in to make the picture clearer, this can provide understanding of cervid dietary responses and ecological interactions in the face of climate change. So that's all from me. I'd just like to thank the conference organisers for putting on this great conference and for allowing me to speak today, and to my institutions and funders down the bottom of the screen. Thank you.